start with a small introduction about our resource person today. Dr. S. Amal has completed his BE in Civil Engineering under Madurai Kamaraj University. Later, he has completed his master's degree in Environmental Engineering, College of Engineering, Guindy. And then he has done his doctoral degree in Environmental Engineering from Faculty of Chemical Engineering, IIT Madras. He has started his career as a lecturer in Thiagaraj College of Engineering, Madurai, and later occupied many positions in many colleges. And he started his profession as an assistant professor in College of Engineering, Guindy, got promoted as associate professor and as a professor in the year 2009. And now currently working as a professor, Center of Environmental Studies, College of Engineering, Guindy. He has a membership in professional bodies like ISTE and IGS. And under his guidance, there are 3 scholars have received their doctoral degrees. And as a joint supervisor, he has guided 1 scholar. And currently, 5 scholars are under his guidance, doing their doctoral degree. And 1 person as a scholar, as a joint supervisor. And he has published 22 international journals and more than 14 national journals. Really very fortunate for us to have such a wonderful person today with our session. Sir, without wasting much of time, we would like to listen to you. Thank you so much. Can you please take over the mic?
Okay, ma'am. Thank you, ma'am. Okay, first, just a minute. I want to share my PowerPoint presentation. That will.
Yeah, now it's okay, sir. Kindly click the hide, show sharing next to that hide is there. No, kindly click that, sir. Otherwise that will be visible.
Just a minute, sir.
Yeah.
Just. So my voice is audible, sir?
Voice is audible, sir. Now your presentation is not visible.
No. Okay, just a minute. I'll take another 1 or 2 minutes. So now PPT is visible, sir?
We could not see the PPT, sir.
Oh, yeah.
Your desktop is seen now. Yeah,
oh, still it's not available. Okay. Just a minute, sir. It's not coming. But, can you permit me to?
No issue, sir.
PowerPoint. How to share it, sir? Your screen, application window, then Chrome tab is there. Which one I have to use to share it?
Entire screen.
Entire screen, but the PPT is already open, but it's not sharing. That's the problem.
So you are minimizing PPT there.
Well, minimize it. Okay.
Your PPT is minimized. Kindly maximize it, sir.
Okay. Then which one I have to maximize?
Your PPT presentation at the bottom, sir.
Okay.
Yeah, okay. We can able to see the screen. Yeah, that is a PPT.
Okay.
Just maximize the screen and you can click F5, sir.
F5.
F5.
Now
Yeah.
it's okay.
Yeah.
Okay.
Now it's okay, sir. Yeah.
Okay. That's
Sir,
right.
You
Sir,
just
kindly close the bottom link, sir. You can able to see hide option is available, sir. The bottom, the blue color.
Blue color is sharing
just click hide.
Okay.
Okay, thank you, sir.
Okay, thank you. Now it is visible.
Yeah, it's visible, sir.
Okay. So very good morning to all present here. So today I would like to brief the various aspects of wastewater treatment, how to treat wastewater and how to produce water for reuse purpose, or it can be let out into water courses. What are the quality standards we have? All these aspects we are going to deal with in an hour. So if we go through the characteristics of domestic wastewater, that is sewage, it contains 99.9% water, 0.1% solids, only 0.1% solids. Out of solids, 70% of the solids are organic in nature, remaining solids are inorganic solids. Mostly organic solids are carbohydrate, proteins, fats, and other things are there. The inorganic constituents are great particles, sand particle, eggshell, broken bones. These are the other composition of inorganic matter, as well as some organic content is also there in great collection. So this is the general characteristics of wastewater. So if you analyze the characteristics of Chennai CMWSSB wastewater, these are the typical characteristics. So suspended solid content will be in the range of 260 to 400 milligram per liter. So mostly the contaminants are spread in three different forms: suspended form, some of the contaminants are in colloidal form, remaining contaminants are in soluble form. So these contaminants present in wastewater or used water can be represented by means of some quality parameters. Mostly suspended solid, the suspended solid spread in water can be represented in terms of milligram per liter, normally 260 to 400 milligram per liter. Every liters of wastewater, it contains 260 to 400 milligram per liter. Okay, then BOD content is very important parameter. The amount of oxygen required to stabilize organic matter under aerobic condition. This is a simple definition of biochemical oxygen demand. Here in BOD experiment, we are trying to degrade organic matter, and organic matter will be transformed into simple, stable, inorganic matter. The main objectives of wastewater treatment is to convert the complex wastewater into simple, stable, inorganic plant nutrient. This is the major objectives of wastewater treatment. The second objective to remove the nutrients present in wastewater. Then final objective to destroy pathogenic organisms present in wastewater. These are the major three objectives of wastewater treatment. The prime objective is to convert the complex nature of wastewater into simple, stable, inorganic matter. Ultimately, our goal is to stabilize the wastewater. So BOD is the critical parameter to evaluate the organic content of wastewater. Normally, it can be conducted in a closed environmental condition by keeping temperature as 27. At the end of third day, we can find out it. In olden days, BOD was measured at the end of 5 days by keeping temperature as 20 degrees centigrade. So nowadays, no need to wait for 5 days. We can measure 5-day BOD at the end of third day by keeping temperature as 27 instead of 20 degrees centigrade. Normally, the BOD content will be in the range of 250 to 400 milligram per liter. What is the meaning of BOD? So 250 means in order to stabilize 1 liter of wastewater, we have to provide 250 milligram of oxygen per liter of wastewater. The maximum saturation DO level in water 9.17 milligram per liter. The temperature is 20 degrees centigrade. This is as per Henry's law. We can arrive saturation value of PV, but the demand is 250. That's why we have to provide more oxygen to stabilize the wastewater. Here in BOD experiment, with the aid of aerobic group of bacteria, we are trying to find out the oxygen demand. So another parameter, chemical oxygen demand. Instead of bacteria, we can utilize the strong oxidizing agent to oxidize all organic matter, biodegradable as well as non-biodegradable organic fraction. So another is the fecal coliform bacteria. These are present in intestinal tract of all warm-blooded animals, human being as well as animal. Then normally it is rated in terms of most probable number per 100 ml of sample. The value may be 10 power 6 to 10 power 7. So total coliform level 10 power 7 to 10 power 8. This is the range we have. So our objective is to keep coliform level, fecal coliform level less than 1000 at the end of treatment. So B-BOD content should be restricted within 20 degrees centigrade if you want to establish any plant in metro cities. So municipalities, the standard is 30. As per Central Pollution Control Board norms, at the end of sewage treatment, the B-BOD content should be restricted within 24 metro cities, if 30 for municipalities and other area. As per suspended solid concentration is concerned, it should be restricted within 50 for metro cities and up to 100 for municipalities. Like that, fecal coliform level should be restricted within 1000. So total general nitrogen is another parameter that is for tertiary standard. As per, so if you want to let out secondary treated wastewater into natural environment, we have to adhere the characteristics of the effluent standard specified by regulatory authority. So wastewater treatment is a combination of unit operations as well as unit processes. In unit operation, we are trying to segregate contaminant with the aid of physical forces. For example, screening operation. The floating solids present in wastewater stream can be segregated by keeping bar screen along the flow path. Another activity, grit removal, as another unit operation. In unit operation, there we are utilizing gravity pull to segregate higher or inorganic solids, mostly inorganic. Some organic solids like food grains also settle during grit settling. So pre-aeration is another. Like that, we have to adopt unit operation as well as processes to segregate contaminants because the contaminants are spread in larger volume. So waste treatment is a phase transfer phenomena. The conservation of mass will not permit us to destroy mass. Here, the mass in one state will be transformed into another state. For example, if organic contaminant is there in wastewater, the organic matter will be transformed into stable inorganic matter at the end of treatment process. For example, carbohydrate is a substance that will be transformed into carbon dioxide, water at the end of aerobic activity. If it is anaerobic activity, it will be converted into methane gas. Like that, the organic matter in solid form will be converted into gaseous form. So waste treatment is the phase transfer phenomena. The mass in unwanted form should be converted into desirable end form. So here the treatment can be attained by adopting unit operations as well as unit processes. In unit operation, we are utilizing physical forces to segregate contaminant. But in process, we are utilizing chemical and biological activity. For example, disinfection phenomena. At the end of treatment, in order to attain the biological quality within 1000, we have to adopt disinfection. Then coagulation, chemical coagulation, advanced oxidation process. These are chemical activity, biological activity, or process. So these activities can be attained with the aid of a reactor. Reactor is a vessel or containment equipped with some machinery. With the help of that, we can attain unit operation or process or both. So wastewater treatment or sewage treatment can be categorized into preliminary treatment. There mostly we are segregating coarse contaminant present in water, followed by primary treatment. There we have settling basin or flotation phenomena to segregate settleable solid. Roughly 60% of the suspended solid can be segregated with the aid of gravity pull. Suppose the specific gravity of the particle is lighter than water, then with the aid of buoyant force, we can segregate solids from aqueous phase. So settling is the solid-liquid separation phenomena. The separation can be done with the aid of gravity pull or buoyant force. So flotation is a typical example for buoyant force. We are utilizing buoyant force to segregate solids. So these are physical operation. Then the secondary processes are mostly biological activity. mostly aerobic for sewage treatment. The sludge can be treated by anaerobic activity. So the various activities we will discuss later on. So the biological solids that will be generated during secondary treatment process can be segregated by providing suitable solid liquid separator at the downstream side we have. Then if you want to reuse the water for flushing of toilets, gardening or industrial use, we should go for advanced wastewater treatment or tertiary treatment. There we have to remove residual carbonaceous matter, the nitrogen content in terms of nitrate or ammoniacal nitrogen, then the excess organic solids, everything should be removed by adapting chemical followed by biological nutrient removal process, then followed disinfection, then RO separation phenomena, reverse osmosis process. By this way, we can reuse the water for industrial purpose. If you want to reuse water for gardening or flushing of toilets, instead of RO, we can provide ultrafiltration modules. These are the various methods available to treat wastewater based on our end use. So this is the scheme we have. So first we have screening activity. Bar screens are provided along the flow path. Here the water is diverted to pass through open channel. By inserting bar, we can remove the floating solids. Then the sand particle, otherwise the heavier solids can be segregated by retaining wastewater in a rectangular type of chamber for a certain time, normally 30 to 60 seconds to remove settlable solid. Then it will be collected in primary settler. There we are trying to remove settlable suspended solids. Then the clarified effluent is further fed into aeration tank. They are converted into biological solids. So by incorporating another solid liquid separator at the downstream side, there's a secondary clarifier. We can collect the biological solids and the part will be redirected back to reactor to keep microbial population within the stipulated range. The excess solids produced during biological system as well as the primary solids, everything will be collected and the solid content can be enhanced by thickening activity. The thickened sludge is fed into anaerobic digester. The solid content of primary sludge may be 1 to 2%, here 0.75 to 1. At the end of thickening, the solid content may be 4 to 6%. The thickened sludge is fed into anaerobic reactor. Here we have two stages. So mostly mesophilic to thermophilic. High temperature is favorable for anaerobic digestion. By this way, we can accelerate the process. At the end of anaerobic process, we can recover energy in the form of biogas. With the help of biogas, we can produce electricity. Whatever energy required to operate the entire system, that can be generated by biogas recovery plant. So most of the conventional system, by incorporating biogas recovery plant, the power required to operate the most of the mechanical units can be, I think 80% of the total energy can be produced with the aid of biogas recovery plant. So this is another scheme we have. The same thing is illustrated here. Here the pumping station be here. The water should be lifted according to the elevation at the disposal point. Accordingly, we have to align all units in such a way to utilize gravity pull. So in designing sewage treatment plant or any industrial effluent plant, the water should be, or the wastewater should be lifted and the subsequent units should be operated under gravity mode. By this way, we can minimize the energy required to operate the system. Otherwise, pumping is required to feed wastewater from one unit to another or one reactor to another. So you should be very careful while preparing the hydraulic flow diagram for the scheme. First, the water should be lifted. Pumping station is mandatory. Then screens are provided to remove the floating solids. Then the grit removal system, oil and grease removal system. If oil and grease content is high, primary settler, biological system, this is activated sludge process we have. The sludge will be collected and it can be thickened, and from the thickened sludge we can recover energy, and the stabilized sludge can be dewatered by providing centrifuge. This is the sludge removal system, the waste treatment system. The effluent from the secondary clarifier can be further be treated based on our reuse standards. So the units that will be added in tertiary treatments will be decided based on the end use. If it is for gardening purpose, we can provide ultrafiltration module. Then again, UV disinfection or ozone disinfection or hypochlorite, any type of disinfection facility to be provided to keep safe. I think the standard to be as per CPHO standard, we have to produce the final product water quality. This is the overall scheme we have. We will deal one by one. The primary treatment units consist of screening facility, grid chamber, followed by primary settler. This is a screening facility. So by incorporating metallic bar along the flow path, we are trying to segregate floating solid. This is a manually cleaned one. This is a mechanically cleaned one. This is a core screen. The spacing 10 to 20 mm here, normally 8 to 12 mm. So spacing is less. Here we can collect the macroparticulate matter floating solids. The other thing, the lighter solids will be collected by providing another fine screen in a sequential manner. The first is a coarse followed by fine. By this way, we can remove the floating solids. Otherwise, these solids may clog the conduits, which is used to transport sewage from one reactor to another. So that's why all floating solids, paper, rags, cardboard, everything should be segregated before feeding water into subsequent treatment. The first unit is screening activity. Screening is a physical action. This is a screen. The metallic bars are provided here. The angle of inclination may be 30 to 60 degree, normally 45. This is the screen facility we have. So this is the screen we have. The angle of inclination, the head loss maximum 30 centimeter. Like that, we can provide some drop here. This is a provision provided to keep the collected screenings. The screenings can be placed here to drain out it, and it can be disposed along with municipal solid waste. For safety relation, we can provide another bypass channel. This is a screening facility. This is a core screen. Core screen we have. Another screening facility is a manually cleaned one. Once the screening activity is over, the water is allowed to pass through channel, and the water will be retained for a time period of 30 to 60 seconds to remove grit particles, a sand particle present in sewage. The sand particle can be collected at the bottom. While designing the system, we have to provide two channels, two parallel channels. One is for cleaning purpose, another is for operation. One is working, another is a standby. This is the manually cleaned screening one. On the mechanically cleaned one, there also we have to provide standby units. So those reactors with less retention time, standby is mandatory. Here the water will be retained for a time period of 30 to 60 seconds. So by keeping water in a stagnated condition, the settlable solid, the heaviest solid, will accumulate at the bottom of the reactor. So that can be collected periodically by manual method or mechanical method. This is a manually operated rectangular type of channel facility to remove sand particle and other heavier inorganic particle. So mostly the characteristics of grit particles 80% or inorganic, 20% organic solid. Some food grains also deposit along with the grit particle. This is another facility, aerated type of grit channel. The purpose of mild aeration is to relieve settling of or relieve any organic solids occur on the surface of the inorganic particle. The purpose of grit collection is only to remove inorganic matter, not the organic solids. That's why the system can be modified by providing aeration facility or the another. Okay, this is the aerated type of grit channels. Mostly we can consider conventional one, that is a better one, because the expensive one we have to provide aeration. This is an aerated type of grit channel. Once the grit removal is over, by providing the partial flow with the ultrasonic way, we can measure the rate of flow. This is a provision provided at the downstream side of grit channel to measure the rate of flow. This is a partial flow with ultrasonic sounding system to measure rate of flow entering into subsequent unit. These are the units. Only first is a screening facility, grit channel we have, then partial flow is provided at the bottom to regulate the rate of flow. At the same time, we can measure the rate of flow entering into subsequent system. Once the screening is over, once the grit removal is over, the wastewater is allowed to pass through primary sedimentation tank. So this is the primary settling tank. Here the water will be retained within a circular chamber for a time period of 2 to 3 years. Normally, the performance of the reactor is rated based on surface loading rate. The rate of volume of water loaded per unit surface area per unit time. The rate of flow is normalized with respect to surface area of the reactor. Settling is directly related to the surface loading, not related to the height of the reactor. Suppose the settling velocity of the particle is greater or equal to surface loading, the particle will be trapped within the reactor. This is the concept behind settling phenomena. Here the water will be retained here. A circular baffle is provided at the center to dissipate the energy. Here, with the end when the water is fed into the reactor in an upward manner, the clarified effluent will be collected by providing weir facility around the periphery. This is a baffle provided here to, to trap any floating solids, oil and grease. So this is a typical diagram of clarifier. It's a circular clarifier. So the clarifier system we have, this is the inlet pipe, so sludgy pipe, the suspended solid. The settlable suspended solid will deposit at the bottom of the reactor, bed slope 1 in 12, height of the reactor, 2.5 to 3.5 is the normal height. Weir facility is provided around the periphery to collect the clarified effluent. This is the effluent collection channel here. At the bottom, we have racker. There is a racker arm. The purpose is to collect the pull sled towards the center. From the center, the sled can be withdrawn by providing sled with travel pipe. This is a wastewater that can be fed into the reactor. This is another diagram we have. This is scrapers are provided at the bottom. So this is the inner circular baffle to minimize the energy. Effluent collection system we have. This is a walkway. The walkway distance here 1.2 meter normally provided here is another reactor. So with this is the effluent collection channel. This is the rotating mechanism. It can be rotated at a speed of 1 revolution for every 30 to 40 minutes. The motor cum reduction gear facility should be provided. Motor cum reduction gear to minimize the RPM of the motor. This can be slowly rotated. That's why the settlable solid that will be accumulated at the bottom of the reactor that can be collected at the center by gravity pull. It can be removed from the reactor because that accordingly we have to align various units. Try to utilize the gravity pull while collecting the residual matter or while feeding water from one reactor to another. Reactor is a vessel. This is another reactor we have. The settling tanks, the reactors are provided, circular baffle, effluent collection system. So once the primary treatment is over, we can remove 30% of BOD present in raw sewage. Suppose the BOD content of raw sewage is 300 milligram per liter. At the end of primary treatment, the BOD content of the clarified effluent is 210 milligram per liter. So if you want to install any plants in metro cities, the effluent standard is 190 milligram per liter of BOD should be removed by biological activity. Mostly the BOD removal is by BOD associated with suspended solid. Roughly 60% of the suspended solid can be captured with the aid of gravity pull. So during primary treatment or pre-treatment and the primary treatment with the aid of physical forces, we are trying to segregate most of the macro contaminant present in water, floating solids, great particles, set level suspended matter. So during primary treatment, BOD removal is BOD associated with settled solids. 60% of the suspended solid, suppose the suspended solid content is 400, 240 milligram per liter of suspended solid can be removed. Remaining unsettlable solid as well as the organic matter in soluble form as well as in colloidal form can further be removed by providing biological system. So biological system mostly aerobic for sewage treatment. The sludge treatment is based on anaerobic activity. So aerobic wastewater treatment. So during aerobic treatment, the complex organic matter, carbonaceous matter, will be transformed into carbon dioxide. So proteins are converted into amino acid, then into ammoniacal nitrogen. Finally, it is oxidized to nitrate by nitrifying bacteria like Nitrosomonas and Nitrobacter. So this is the end product. So during biological activity, the complex organic matter will be transformed into simple stable inorganic matter. So during oxidation phenomena, a lot of energy will be liberated. So with the help of the energy, bacteria produce more and more bacterial cell by binary fission. This is a representation of bacteria. Roughly 1 kg of C-BOD is converted into 50% of biological mass. The aerobic process, we require molecular oxygen. Here, molecular oxygen will act as a terminal electron acceptor. During oxidation phenomena, electrons are liberated. In order to accept those free electrons, so we have to provide appropriate terminal electron acceptor. In aerobic process, molecular oxygen will behave like a terminal electron acceptor. With the help of electron acceptor or based on electron acceptor, we can categorize biological activity into aerobic process, anaerobic process, and anoxic process. Anoxic process, nitrate will act as a terminal electron acceptor. Nitrate is reduced to nitrate, then denitrification phenomena. Another group of bacteria, facultative bacteria, they can survive in the presence or absence of oxygen. But aerobic process, molecular oxygen is mandatory. The system should be provided with adequate quantity of oxygen to oxidize all organic matter present in water. The maximum saturation level very close to 9 at 20 degrees centigrade in TDS for free water. How do we provide 210 is the remaining? If the raw B-word is 300, so 20 is the standard, then 190 should be removed. How do you remove that one? Suppose the capacity of the plant is 100, we can easily calculate the BOD load entering into the reactor. If it is aerobic process, for every kg BOD entering into the reactor, we have to provide 0.8 to 1.1 for conventional ASP system. So that much oxygen should be provided. How do we provide that much oxygen? So we have to indirectly accelerate the mass transfer phenomena by agitating the fluid. That is an indirect way of mass transfer. Another is the diffuser aeration system. Air can be directly sponged through the reactor. It's an aerobic activity. The another process, anaerobic system. Anaerobic bacteria can survive in the absence of oxygen. The intermediate that will be generated during anaerobic pathway will act as a terminal electron acceptor. So no need to provide external source of terminal electron acceptor. The intermediate that will be generated during anaerobic pathway will act as a terminal electron acceptor. Here the polysaccharides like carbohydrate, protein, the polysaccharides are converted into simple monomers or monosaccharides by enzyme-mediated transformation. Any biological system, enzymes are mandatory. So mostly they are secreted in outside of the bacterial cell. Their role is to convert the insoluble organic matter into simple, stable, organic monomeric substance like carbohydrate into simple sugar. Cellulose will be converted into simple sugar. Proteins are converted into amino acid. Like that, the insoluble organic matter will be converted into soluble matter. So in anaerobic activity, enzymatic transformation is the limiting aspect. While designing any anaerobic system, we should know the limiting aspect of the reactor. In anaerobic activity, mostly hydrolysis, that is the enzymatic transformation, that is the hydrolytic enzymatic action is the limiting phenomenon. Like that, once you know the limiting aspect, then we can easily solve the problem. So then second stage, the monomeric substances are converted into simple low boiling organic acid by fermentive bacteria, acid producing bacteria. Then the butyric and the propionic acids are further converted into acetate by acetogenic bacteria. Then finally, two different types of methanogenic bacteria. One is hydrogen utilizing bacteria, and another is acetate utilizing bacteria into methane and carbon dioxide. In anaerobic activity, we can recover biogas. Energy recovery is only possible during anaerobic activity. For every meter cube of biogas, we can definitely, of methane content 75%, we can definitely produce 1.4 units of electricity. It's a very important aspect in waste treatment. Energy recovery, most of the wastewater treatment facility, we can produce our own energy by incorporating anaerobic system, sludge biogas recovery system. This is the basis of anaerobic system, anaerobic process. So I would like to compare the performance of aerobic and anaerobic process. During aerobic process, the organic content will be converted to sludge. 40 to 60% of the organic matter will be transformed into sludge and carbon dioxide and water. But in anaerobic activity, the volatile fraction is converted into methane gas, carbon dioxide. Lesser quantity, 5 to 10% of the organic content is converted into biosolids. During aerobic treatment, most of the solids are converted to biosolids. That's why solid liquid separator should be incorporated at the downstream side of biological system. So this is the various stages in biological activity. I would like to skip here. So these are the various reactors. We should know the reactor. What do you mean by reactor? I have already pointed out reactor is a vessel or containment equipped with some machinery. With the help of that, we can attain unit operations or process or both. Here is a reactor can be categorized into batch reactor and a continuous flow reactor. Continuous flow reactor can further subdivide it into completely mixed flow reactor or continuous stir tank reactor and a plug flow reactor. In completely mixed flow reactor, so there is no spatial gradient within the system. The microbial population as well as the substrate, the BOD content is uniform throughout the reactor. There is no spatial variation. But here, in the outlet concentration equal to reactor constant. These are the peculiar characteristics of completely mixed flow reactor. The plug flow reactor is the piston flow first in, first out. The packed bed system, while developing mathematical model for packed bed system, we can consider the reactor as a plug flow model. So if you want to develop water quality model for river ecosystem, then it will be treated as a plug flow regime. Like that, we can develop model for any system. So for today's discussion, I have considered 6 options. So what are the options available to treat wastewater? First is the activated sludge process, conventional one and extended aeration. Second one is the moving bed biofilm reactor. This is the modified version of ASP. Then the aerated biofluidized bed reactor. This is another version we have. Then third option, we have sequential batch reactor. So with the aid of a single vessel, all activities are taking place in a sequential manner. Membrane bioreactor is the new development. There, ultrafiltration module will act as a solid liquid separator. In conventional ASP system, as well as moving bed biofilm reactor, settler, gravity settler will act as a solid liquid separator. Option number 5, upflow anaerobic sludge blanket reactor, followed by suitable aerobic system. By this way, we can treat wastewater. Trickling filter system is an outdated system because the efficiency of the system is very, very low. But here, ASP system above 90%, moving bed biofilm reactor 95%, extended aeration above 98%, sequential batch reactor above 95%. Here, the trickling filter only 80 to 85. 80 is also the difficult task here. ASP system 75 to 80 only. So this is the conventional ASP system. ASP system consists of the primary treatments, pre-treatment facility, the pre-treatment and the primary treatment. The primary clarified effluent is loaded into aeration tank, followed by a secondary settler. This is a typical setup. So this is the flow diagram for conventional ASP. ASP means activated sludge process. So once the pre-treatment is over, we have to provide primary clarifier to collect the settleable solid. Then primary clarified effluent is loaded into aeration tank. So air is poured through the reactor by providing diffuser tube at the bottom of the reactor. Here the organic matter is transformed into biological solids. Everything will be collected in secondary settler. With the help of gravity pull, we can remove the biological solids and it can be treated by providing sludge handling system. Thickener, anaerobic stabilization phenomena, energy recovery option. Then the stabilized sludge can be dewatered by providing sludge drying bed or centrifuge, mechanized system or natural system to treat sludge. This is the process flow diagram for conventional ASP system. This is the aerobic process, is aeration tank followed by secondary settler. The part of the activated sludge is redirected back to the reactor to keep bacterial population within the stipulated range. This is the aeration tank we have. This is a mixer liquor suspended solid. This is a mixture of incoming primary effluent and the return activated sludge. The solid content may be 3000 to maximum 3000, 1500 to 3000 milligram per liter. So this is the mixer liquor suspended solid, MLSS. The volatile fraction may be 75 to 85%, 80% volatile fraction. This is a flux. While feeding wastewater into acclimatized culture, the organic matter is transformed into biological solid. This is a biological solid, so bacteria. So in flocculent form, and it's flux only. This flux can easily be segregated with the aid of gravity pull. So during biological activity, the organic matter will serve as food for aerobic bacteria. The aerobic bacteria utilize their energy or produce their energy with the help of organic matter, and they reproduce by binary fission. And so within a short duration, plenty of cells will be formed and they can be joined together form a bigger mass, and it can be easily be segregated by gravity pull. This is another facility because of mechanical agitation. Instead of providing diffuser tube at the bottom of the reactor, we can indirectly accelerate the mass transfer phenomena by spreading water into larger area by vigorously agitating the fluid. During agitation, we are trying to create tiny liquid droplets because the mass transfer will occur through the interface only. Oxygen in atmospheric air will be transported into liquid phase through interface only. So mass transfer rate can be accelerated by creating turbulence in liquid phase. So another thing, we have to create more surface area. By this way, definitely or indirectly, we are trying to accelerate the mass transfer phenomena. One is directly we can spot air at the bottom of the reactor by providing diffuser tube. Otherwise, we can indirectly accelerating mass transfer rate by spreading water into larger area by agitating the fluid with the aid of mechanical paddle. Here at the downstream side, we have secondary settler, and the part of the biological solids or bacterial solids will be segregated and it can be redirected to the reactor. So this is a blower facility. Air blowers are required to supply air to the reactor. So this is oxygen requirement. Oxygen requirement can be quantified based on rate of flow. Suppose the capacity of the plant is 100 milliliters per day. Q. S0 is the influent BOD. Influent BOD 210. Effluent BOD is the standard 190. Okay, we can easily find out. Px is the net quantity of biological solids that will be formed during aerobic transformation. The oxygen equivalent is 1.42. So by this way, we can quantify oxygen required for biochemical activity. F is the conversion factor to convert BOD3 to ultimate BOD. These are the basic calculation. So based on the influent BOD level and the desirable effluent standard and the rate of flow, we are trying to quantify how much oxygen is required for carbonaceous removal. If it is protein, we have to calculate for every kg of TK and total general nitrogen, we have to provide 4.6 kg of oxygen. So if you want to attain nitrification in addition to carbonaceous removal, we have to provide additional oxygen requirement. So okay, while providing diffuser tubes at the bottom of the reactor, we have to consider the mass transfer constraint because based on the location, oxygen transfer varies with respect to based on location as well as the salinity factor. Surface tension is another factor influencing the mass transfer rate that can be incorporated in terms of beta. Surface tension factor, salinity and surface tension correction factor beta 0.95. Saturation DO level with respect to altitude, temperature as well as altitude. We have to consider this is for sewage. So based on the sewage temperature as well as the location of the plant, we have to find out the DO level. This operating DO level 1.5 to 2 milligram per liter. DO saturation of tap water 20 degrees centigrade 9.17. 1.024 is the temperature correction coefficient. 20 is the benchmark. T is the temperature of wastewater. Alpha is the mass transfer correction factor. F is a fouling factor. By incorporating all mass transfer constraints, we have to work out the standard oxygen transfer requirement or standard oxygen transfer efficiency. So based on that, we have to quantify how much oxygen required as per field condition. So based on that, we have to provide diffuser tube at the bottom of the reactor. These are the diffuser tube. The length of the tube 1 meter, diameter 90 millimeter. I think the capacity of the diffuser tube 10 metric cube of air per hour. 10 metric cube per hour. So oxygen transfer efficiency 6% per meter depth. The depth is 5 meter. The clearance is 24.58 is the height of the liquid, actual height available. So 5 meter is the height of the reactor. The clearance, bottom clearance 20 centimeter, 4.8 into 6 by 100. Once you know the oxygen content of oxygen content in air, so 2.21% is a volumetric proportion, 22 points fraction, or you can say 23 for mass fraction. All aspects we have to consider while quantifying the how many diffuser tubes are required to perform this phenomena. So how many the diffuser tube or the number of tubes can be decided based on theoretical prediction based on the quality as well as the quantity of water to be treated. So based on the manufacturer's specification, we have to design that one. So nowadays up to 7.5% transfer efficiency diffuser tubes are available. This is a diffuser tube we have. Aeration tank with the diffuser tube at the bottom of the reactor. This is the diffuser tube, a membrane type. This is the air from this is connected to air blower. So air blower, this is connected to air blower. These are the diffuser tube. Length 1 meter, diameter 90 millimeter. Capacity 10 meter cube of air per hour. So based on the demand, we have to supply as well as the oxygen transfer efficiency. Oxygen transfer efficiency, fine diffuser 6% per meter depth. 6% per meter. Accordingly, we have to design the mechanical system. This is a diffuser tube we have. This is a diffuser tube. This is another setup with the diffuser tube at the bottom of the reactor. This is the bubble. Okay, this is a fine bubbles. We have to create fine bubble with the aid of a membrane type of diffuser tube. This is a length is 1 meter, diameter 90 millimeter. Air is forged through the reactor. So this is the reactor during working condition. Here we are keeping the solids in under suspended state. So we are not allowing the solids to settle at the bottom of the reactor, and everything will be collected in this another view for mechanical agitation. This is MLSS we have. This is aerator. This is surface aerator we have. So once the activity is over, once the aeration is over, everything will be collected in secondary settler, and the pot will be redirected back to the reactors. The design is very simple. So based on the load, we have to design the mechanical system. The fluid will be retained for a time period of 4 to 6 hours. Normally 4 to 6 hours. Accordingly, we have to size our reactor. So once this is another system, because the modified version of activated sludge process, extended aeration process. So most of the new plants are extended aeration type process. No need to provide primary clarifier. So pre-treatment is more than enough to remove sand particle as well as the floating solids. Here the MLSS microbial population is up to 4,500 milligram per liter. In addition to carbon removal, we can attain nitrification by operating our system at low organic loading rate. So that is another condition we have. Here we have to provide lesser food and more microbial mass. We are trying to keep the reactor at endogenous phase of respiration by providing lesser food, lesser organic matter, and more microbial mass. By this way, the solids, we are allowing this biosolids entering into endogenous phase of respiration. Most of the solids are completely stabilized. Partial stabilization will occur. Here we are retaining biological solids up to 24 days. As per CPHO guidelines, 10 to 24 days. So liquid will retain up to 24 hours. Okay, then we have secondary settler. For secondary settler for solid liquid separator, and most of the biological solids can be redirected back to the reactor. Excess solids will be collected in sludge thickener. This is the gravity type of reactor, and the thickened sludge is further dewatered by providing sludgy drying. But this is the scheme we have. One is first scheme, conventional ASP or extended aeration system. This is another one, oxidation. This is another setup we have. This is another example for extended aeration system. This is a bacterial biological mass. This is a mechanical facility. It's a cage rotor. It's a horizontal type of aeration facility. It's an oxidation, which is another one. It's an outdated system because the space requirement is huge. But this is another system we have. Next system, moving wet biofilm reactor, MBBR. MBBR version is the different version. In addition to suspended growth, here we are providing some attached growth. So wastewater treatment can be attained by suspending microbial species in a vessel or in a reactor, or it can be immobilized or attached on the surface of the solid media. Here we are trying to integrate attached growth culture with suspended processes to keep microbial mass for longer time, to retain microbial mass for longer time. Here biomass holdup is high. Any biological system, the relative rate of mass removal. The relative rate of mass removal is related to internal and external mass transfer rate and the microbial mass present within the system. To provide more microbial mass, more food will be assimilated. The unwanted contaminant or the organic matter will serve as food for bacteria. We are trying to utilize bacteria to degrade the organic matter, and the complex organic matter, the polysaccharides, are converted into simple stable matter. So by this way, we are trying to stabilize the wastewater. That is our ultimate aim. Another objective, we are trying to remove the nutrients in the form of proteins. Protein is the major source. Protein is converted into ammoniacal nitrogen during ammonification process. It is further oxidized to nitrate during nitrification phenomena. It can be reduced to nitrogen by denitrification activity. Here, nutrient removal is somehow possible, not complete removal, only nitrification is possible. Combined carbon oxidation as well as nitrification can be attained by providing space for nitrifying bacteria along with carbon oxidizing bacteria. Two different types of bacteria coexist in a single reactor. All other components are more or less similar to conventional ASP system. Only thing, we are providing some attached growth. This is the media pickup. So 40% of the liquid volume is occupied by media. 25 to 40 is the normal range. Sometimes it can be 50, normally 25 to 40. 40% of the media volume is occupied, reactor volume is occupied by plastic film. This is the media with biological slimy layer around the surface of the media. So the specific surface area 200 to 400 meters square per meter cube. Porosity above 95%. Specific gravity, they are lighter than water, very close to water, mostly lighter than water. This is the characteristics of the packing media, mostly plastic films, PVC films. This is the reactor, moving bed biofilm reactor. Here we are providing space for attached growth as well as suspended growth. So part of the organisms are under suspension. Remaining organisms are attached or immobilized on the surface of the packing media. By this way, we can provide more biomass holdup. Biomass means bacteria. Biomass holdup. And in addition to carbon removal, we can attain nitrification. These are the media available in market. Any size we can procure from the market. These are the different orientation of the media, different configuration of the media. This is the reactor with the media. The black spot representing the media. The other arrangements or everything are more or less similar to conventional ASP system, only in aeration tank. In addition to suspended growth, part of the microbial species are immobilized on the surface of the packing media. This is a reactor provided with packing media for attached growth. Here we are trying to integrate two activities. One is suspended growth, another is the attached growth. By attaining, the purpose is to produce higher quality effluent. By this way, we can definitely produce B.O.D. less than 10 milligram per liter. Definitely better quality. Nitrification is possible. Complete nitrification is possible. Sludge stabilization is also possible. Okay, this is another system. This is a fab reactor. This is a fluidized bed reactor. The same as the MBBR configuration. Only the packing media is fluidized in liquid. By this way, we can create more mass transfer, and the carrier will move throughout the reactor and utilize substrate throughout the reactor. There's only more or less thing. There is no much difference between MBBR. Only there's the representation purpose. What is fluidized bed or moving bed biofilm reactor? Next activity is very important. So nowadays, 80% of the new plants are SPR based plant. 80% of them. So most of the new plants in Chennai are SPR based. In future, SPR is a popular one. SPR nowadays is a popular technology. SPR means, I think this is a modified version of extended aeration system, but all activities are taking place in a single vessel in a cyclic manner instead of providing multiple vessels in sequence. Here, pre-treatment is mandatory. Once the pre-treatment is over, the effluent is loaded into SPR basin. Okay, then we have to allocate time. So filling phase, reaction phase, settling, decondition, idle. All these activities are taking place in a sequential way in a single reactor rather than providing multiple reactors in sequence. So no need to provide a number of reactors to attain all these activities. Biological activity as well as solid liquid separation can be attained in a single vessel. Minimum 2, maximum 4 or 6, 8, any numbers we can provide. At least 2 is required, because at a particular time, at least one operation should be going on at least one reactor. Like that, we have to design. The system can be operated by PLC controller or program logic control. So no need to operate the system under manual method. This is a system because the decanter is provided to collect the clarified effluent. At the downstream side, we have the sludge removal, sludge treatment facility, and the treated effluent can be further used for flushing of toilets by disinfection. So before disinfection, we can have some fine-tuning process, ACF filter, PSF unit to remove the residual suspended solid, and it can be disinfected, or we can select ultrafiltration module, then disinfection, and it can be used for flushing of toilets. Okay. This is the phase. SPR basin consists of various phases: filling of wastewater, then aeration, at least 2 hours. Remaining other 2 hours can be totally 4. Normally 4 to 6 cycles per day. 4 to 6 cycles per day. So filling phase, half an hour filling, 2 hours maybe aeration, half an hour for settling, half an hour for decondition, idle sometime. Like that, the 4 to 6 hours is the total reaction times for one cycle. One cycle, 4 activity, 5 activities are there. During idle, we can remove the excess lead, or it can be done along with during decondition. We can avoid idle operation. So here in the sequential batch reactor is a modified version of extended aeration system. Here, all activities, all active physical as well as biological activities are taken. Physical is a filling is a physical, decondition is a physical activity, settling the gravity. All activities are taking place in a sequential manner by providing a single vessel rather than providing multiple vessels in series. The very important aspect of this reactor is actual plants. There's totally 6 numbers of reactor we have. At a particular time, one of here aeration is going on. Here the decondition is going on. Decondition is almost over here. Then here the filling operation is going on. Here this is a decanter. The clarified effluent can be collected with the aid of decanter. Okay, so we have to keep 2/3 of the remaining MLSs for subsequent cycle. No need to provide another solid liquid separator at the downstream site to segregate biological solids. Here, biological activity as well as solid liquid separation can be attained with the aid of a single vessel or a single reactor. So this is another reactor. Aeration activity is going on here. The SBR tank, so at the end of decondition. Once the decondition is over, we have to fill the new batch of wastewater. Decondition, then idle. So filling, aeration, then settling phenomena of another decondition, removal of clarified effluent by lowering the decanter. There can be mechanical facility we have. Then the idle operation, we can withdraw. It's like rate at which solids withdrawn from the reactor equal to rate at which solids generated from the reactor. This is a control mechanism we have. So everything can be controlled by providing suitable control mechanism. So this is the next system. So membrane bioreactor. So membrane bioreactor is another type of system we have. Here, the ultrafiltration module will act as a solid liquid separator. In ASP system as well as MBBR system, moving bed biofilm reactor system, there we have to provide secondary settler or gravity settler at the downstream site to segregate biological solids. In SBR basin, the SBR basin itself will act as a solid liquid separator. There no need to provide additional solid liquid separator. Here, instead of gravity separation, we can incorporate ultrafiltration module or microfiltration, mostly ultrafiltration. So ultrafiltration module trap the suspended matter. So that can be provided at the side stream, or it can be provided in a submerged mode. So submerged mode are more popular now. So 2 modes we have. The settler or the separation unit can be provided at the downstream side. That says, okay, side stream 1, side stream, or it can be inside the aeration basin, submerged type of MBR. MBR means membrane bioreactor. MBBR means moving bed biofilm reactor. ASP, activated sludge process. This is the MBR system. Other systems are more or less common. So raw sewage collection well. So this is storage well. So 30 to 60 seconds can be provided to minimize the velocity and can allow to pass through the channel. Bar screens are provided. Grit removal mechanism we have. Then it is loaded here. No need to provide primary clarifier. Here the MBR tank we have. Membrane bioreactors. This is the NDF system is a membrane bioreactor. Here the ultrafiltration module will act as the solid liquid separator. That is the only difference we have. Sludge drying bed, sludge handling system we have. But here we can keep microbial concentration up to 12,000. So practically 8,000 to 12,000, but theoretically up to 20,000 is there. But the practical value up to 12,000. Conventional ASP 3,000 milligram per liter microbial mass, but the extended aeration up to 4,500. Here 12,000 is possible. More microbial mass. Here we can produce the final product less than single digit. BOD or suspended matter, everything is less than single digits. Better quality, excellent quality we can produce, but the cost involved in the treatment scheme is very, very high. This is a system we have. So if you want to remove mutagens in addition to carbon, we can provide anoxic tank. Anoxic means the nitrate is transformed into nitrogen gas with the aid of denitrifying bacteria. So denitrifying bacteria utilize nitrate as the terminal electron acceptor. That's why we have to provide a pump here. So during aerobic activity, The proteins are converted into ammoniacal nitrogen, then into nitrate. Nitrate will be collected here. The partially nitrified effluent contains nitrite, everything will be recirculated back to the anoxic tank. The organic carbon will act as a carbon source for denitrifying bacteria, and it will be converted into nitrogen. So by providing scheme in such a manner, we can eliminate nitrogen from wastewater. So nitrogen removal is also possible in the MBR reactor, membrane bioreactor. So anoxic tank, we can provide this a pre-anoxic tank that is provided prior to biological activity. Here we have aerobic tank. Here the diffuser aeration system is provided. The ultrafiltration module will act as a solid liquid separator, permeate as a excess lead can be collected from the reactor and it can be further treated. This is a schematic representation of MBR system, membrane bioreactor, pre-anoxic tank, aerobic tank, and a solid liquid separator that can be the submerged type of reactor. The ultrafiltration module is incorporated within the aeration tank. This is a submerged type of MBR. Another is a side stream MBR. This is okay. Another is a submerged one. So membrane is provided within the reactor. This is the unit we have. Aeration tank with ultrafiltration module. UF module is provided here. These are the solid liquid stuff. We can collect the permeate here. So from the membrane, we can collect the permeate. The solid content is up to 12,000 milligram per liter. 12,000 milligram per liter, 1.2%. So very huge content. But conventional ASP, we can attain up to 3,000. Extended aeration 4,500 is possible. Here, more microbial mass, the quality is excellent. We can produce single-digit effluent quality parameter. But the pathogens are there in finally treated water. It can be destroyed by providing suitable disinfection facility. So MBR treatment is a very sophisticated system, but the operational cost will be high. So investment cost is also high. We have to provide pumping system, high pressurized pump to pull the solvent towards the membrane. So membrane separation phenomenon is a pressure-driven process. This is a membrane normally used for MBR reactors. MBR is I would like to emphasize the difference between conventional ASP as well as the first generation MBR as well as second generation. First generation MBR, the ultrafiltration module is attached at the downstream side of aeration tank. But in second generation MBR system, the ultrafiltration module is provided within the aeration tank. In conventional ASP system, we have gravity separator to segregate the biological mass that is provided at the downstream side of aeration tank. But in MBR system, membrane bioreactor, the ultrafiltration module, here instead of gravity separator, we have ultrafiltration module. That is the only difference between conventional ASP and the MBR. So far, we have discussed conventional ASP, extended aeration system, moving bed biofilm reactor is an integrated reactor. So the fourth one is the membrane bioreactor. Membrane bioreactor, the ultrafiltration module will act as a solid liquid separator. In between, we have discussed about the SPR reactor, sequencing batch. That is a modified version of extended aeration system, but all activities are taking place in a cyclic manner. It's a batch type of reactor, cyclic process, cyclic extended aeration activated set process. This is the MBR. It's actual plants, actual MBR plant. Membrane, these are the membrane. So not this is the membrane. This is the purpose is only to highlight it. This is to highlight the membrane. Next system, we have UASP reactor, upflow anaerobic sludge blanket reactor. This is an anaerobic system. So in the anaerobic system, we have to, the anaerobic system will act as a primary treatment system, followed by some polishing treatment is mandated because the maximum possible BOD removal 75 only. 75 is possible. Suppose the BOD content of raw sewage is 300. At the end of anaerobic treatment, the BOD content is how much? There is 75 milligram per liter BOD is there. Okay, the standard is 20. In order to attain the stringent discharge standard, we have to incorporate suitable downstream side treatment system for UASP. So if we consider, but this energy cost is very, very minimum. So if you operate your system under gravity mode, there is no energy cost involved in UASP system. Only the wastewater should be fed through the reactor by gravity. It's a gravity one or pumping mode. So based on the topography, pumping or gravity can be adapted while erecting the reactors. A primary treatment facility, it can be lifted and all other downstream side treatment schemes can be align according to the topography of the land. By this way, try to minimize the cost of the operational cost. While designing any treatment scheme, we should focus on operational cost as well as the energy cost. That is very important thing. The energy cost can be minimized by aligning the reactor based on the topography. Accordingly, we have to prepare alternative design, and the better one will be considered for our final. Okay. So this is a USP reactor. At the downstream, we have aeration system, then secondary clarifier, disinfection unit. The slide, the excess slide from USP reactor as well as the biological solids will be thickened and it can be dewatered. So if we select extended aeration and USP system, no need to provide another anaerobic digester here. Gas can be recovered here, and from that we can generate electricity. From the USP system, electricity can be generated because the anaerobic reactor. Okay, energy recovery is possible. Operational cost is minimum. The initial investment cost is also low, but the land requirement is somehow high when compared to SPR and the MBBR system. So this is the sequence involved in USP treatment. So if we consider USP as a major biological treatment system, so we have to align various units by this fashion only. This is the USP reactor upflow anaerobic sludge blanket reactor. It's an anaerobic system. It's an upflow type of reactor. The wastewater is loaded at the bottom by providing inlet pipe for every 4 square meter floor area. This is a guideline we have to follow while designing the reactor. For every 4 square meter floor area, 1 inlet pipe should be provided. By this way, we can disperse wastewater evenly at the bottom of the reactor. Okay, short circuiting should be avoided. The feed water should not leave along with treated air, along with the effluent without undergoing any treatment. That's a short circuiting. So we have to minimize the short circuiting by dispersing or wastewater evenly by providing 1 inlet pipe for every 4 square meter floor area. So this is the basic guideline we have to follow. In addition to that, the minimum COD load 1 to 2 kg COD for every meter cube per day is a minimum requirement. So retention time up to 10 hours for average flow. So peak maximum 4, not 4 to 5. These are the guidelines we have to follow while designing USP system for sewage treatment. This is the polishing treatment provided at the downstream side. This is an aerated type of lagoon, or any extended system can be provided to prepare product water in line with CPCB standards, Central Pollution Control Board norms. Okay, we have to, this is a reactor because this is an inlet pipe, a feeding pipe. For every 4 square meter floor area, 1 inlet pipe should be provided. This is a gas collection tube. This portion will act as a solid liquid separator. No need to provide solid liquid separator for the system. The gas will be collected from the top of the reactor. This is the upflow type of reactor. That's why the reactor is called upflow anaerobic sludge blanket reactor. This is the anaerobic system. We can tap biogas. Energy recovery is only possible here, only possible during anaerobic activity. This is a system because this is a deflector beam that is during construction phase. This is a reactor. The last one is the trickling filter. So trickling filter is the attached closed system. It's not a popular system because the maximum possible BOD removal 80 only. 85 is not theoretically 85, but practically 80 only. So by providing 2-stage filter, we can somehow attain 90% of efficiency. But the space required to construct the scheme is huge. As well as water nuisance are also there. It's only outdated system. But anyway, you should know the configuration of the system. We have the pre-treatment facility, then a primary settler, trickling filter, so followed by aeration. This is a modified version we have, but it's not applicable. But it's not, practically it's not at all possible to produce the anticipated effluent quality by trickling filter system. Here, the organisms are immobilized on the surface of the media. So these are the stages of biofilm development. So bacterial cell excretes some kind of polymeric substance. This is a glycocalyx, this strands. So then they form a slimy layer around the surface of the cell. This is the actual biological film. This is the bacteria. So bacteria, single cell. The flagella is there. So this is a bacterial cell. These are the polymeric substance, glycocalyx. This will aggregate the loose cells and everything will be adhered on the surface of the packing media. So the attachment is based on the glycocalyx. This is some pictures, scanning electron microscopy of immobilized microbial culture. This is the bacterial cell, the spherical shaped cell. This is another cell, it's a very interesting cell. Okay, this is the working principle of trickling filter, the wastewater is sprinkled on the top surface of the media. Through the trickling filter, the under drainage is provided at the bottom to collect the percolate. This is a reactor beaker. Here the wastewater is the gravel is used as a stone, as a packing media. That is not normally adapted for low rate trickling filter. Here we can load up to 1 to 4 meter cube per square meter per day. It's a low rate trickling filter, stone as a packing media. The maximum height 2.5 meter. The ventilating ports are provided at various locations to ensure better mass transfer rate. The shiny layer represents the microbial mass. The microbial mass will be established around the surface of the media. So basically, once you sprinkle wastewater on the top surface of the reactor, the organic matter present in liquid will diffuse through the biological film. Then it will be stabilized by aerobic bacteria. The stabilization is by diffusion followed by biochemical oxidation. Diffusion is a transport phenomena. Biochemical oxidation is a combination of chemical oxidation and as well as biological activity. This is another reactor. This is a stone. This is a pebble is used as a packing media. The wastewater is sprinkled evenly throughout the reactor by providing distributor arm. This is a reactor. This is another reactor with the plastic fill as a packing media. So plastic fill as a, this is a plastic modular sheet plastic media. The voidage is high because the oxygen is transported through the pore space only. The surface area available for biofilm growth is in the range of 200 to 300 meters square per meter cube. So a lot of space, the surface is available on the surface of the packing media. So this is another type of packing media, normally used for cooling tower. But here we can use as a packing media for trickling filter. This is a manufacturer's Mundus. The angle of inclination 45. This is the hydrodynamics of the fluid, so how the liquid is flowing. So how to create mass transfer where liquid is splitted as well as joining at the nodal point. So this is the top surface of the media. Here this is a trickling filter. The wastewater is evenly sprinkled on the top surface of the reactor by providing distributor arm. So this is the distributor arm. The percolate can be collected by providing under drain at the bottom of the reactor. This is the specification of trickling filter, but it's outdated system. High rate trickling filter also, the space requirement is also huge. So another system is a natural based sewage treatment system, it's a pond system. So far we have discussed various mechanized system, conventional ASP, extended aeration ASP system, moving bed biofilm reactors, the integration of suspended growth and attached growth, sequencing batch reactor. All activities are taking place in a sequential manner instead of providing multiple reactors in sequence SPR mode. MBR is a membrane bioreactor. There, the ultrafiltration module will act as a solid liquid separator. Then we have USP reactor followed by aerobic polishing treatment. Then trickling filter is attached to the system. Efficiency is very, very low. The space requirement is also huge. It will be treated as outdating treatment. The final treatment is the natural based treatment system. There is no mechanical facilities, but earlier system we have the mechanical facilities. Pond system, aerobic pond. So ponds can be categorized into aerobic, facultative, and anaerobic pond. Aerobic and facultative ponds are working based on algal bacterial symbiosis because algal cell produce oxygen during photosynthetic action. This oxygen will act as the terminal electron acceptor for bacteria. So bacteria utilize the organic matter and it will be stabilized into carbon dioxide, ammonia, or nitrate. And during aerobic transformation, the organic matter, the energy will be utilized by bacteria to produce more bacterial cell. This is a symbiotic interaction. So both these two species are immensely benefited for their mutual interaction. So algae, with the aid of sunlight, they can produce their own food energy. So during photosynthetic action, oxygen will be liberated. That oxygen will serve as a terminal electron acceptor for aerobic bacteria. Aerobic bacteria stabilize the organic matter by aerobic stabilization phenomenon. This is a symbiotic interaction. The pond system we have that is illustrated here. The pond is only these are the various layers of pond. Aerobic pond, we can is another pond, the actual pond system. In order to avoid groundwater contamination, the bed as well as site should be properly lined. So lined system we have. So based on the soil permeability, we have to select suitable liner systems. So liner means we can protect the underground water. Otherwise, the quality of underground water near adjoining area may be highly affected. That can be avoided. This is the actual scheme. based stabilization pond, but the wastewater will be stored up to 15 days. Normally, ponds are connected in series: anaerobic pond, facultative pond, aerobic pond, maturation pond. Like that, we have to connect ponds in series. First, we can have anaerobic pond. So fully, the system is free from oxygen. The system is completely devoid of oxygen, followed by facultative pond we have. Once the anaerobic activity is over, it can be loaded into facultative pond. Then this is the final, this is the aerobic pond we have. This is the aerobic pond. It's a maturation pond. So like that, ponds are connected in sequence. Anaerobic, facultative, aerobic, and the final pond is a fishing pond or maturation pond. So here the disinfection can be done with the solar energy, and the water can be used for irrigation purpose. This is the low-cost treatment system. If the land space is surplus, then we can consider this WSP as an alternate technology when compared to mechanized system. This is another scheme we have. This is a cross section. Here we can compare the quality of water, how to produce. First, we have ASP and extended aeration combination. We have ASP, extended aeration system, MBBR system, MBR system. Final system, we have SBR system. Okay, how to prepare the quality of effluent, so what type of treatment is required, capital cost, chemical cost, maintenance cost, everything can be compared here. And the various treatment facilities, everything is consolidated here. For example, if we consider conventional ASP, for every kg BOD, we have to provide 0.8 to 1 kg of oxygen. Extended aeration slightly above. Here we are allowing nitrification. That's why carbon demand, oxygen required for carbonaceous oxygen demand, and the nitrification demand. Everything will be accommodated here. SPR also same one. The MBBR supposed aeration. There's another thing we have. These are the specification we have. So when compared to land area requirement, USP system, we need more area. Second one is ASP. Like that, other systems are like that. So MBR system is a space-saving one. MBR and the SPR system. So within the limited space, we can accommodate all facilities. But the investment cost is huge for USP system. USP system investment cost is huge. So post-treatment is the required. Let's say the cost is huge. Okay, the capital cost of the system is USP. MBR is high. MBR USP is low. Cost is high, but the cost of total capital cost of the MBR, this is a membrane system we have, that is high. The maintenance cost, this is another thing, is the power cost. The power required for MBR is also high. The pumping system we have. Okay, the investment cost is huge for MBR. Then second thing is SPR. We have decanter facility. All other system, there is no much difference. So MBR, the power, the capital investment cost is high, as well as the power requirement is also high. This is the maintenance cost. Maintenance cost of MBR, because we have membrane. Once in 3 years, we have to replace the membrane. A lot of difficulties we have in MBR system. The membrane driven process, we have to investment huge money, as well as the maintenance cost is high. But here we can produce the excellent effluent quality. The effluent quality is mentioned here with respect to system. We can USP reactor inferior quality. Then second ASP, then MBBR, then MBR and SPR, we can produce excellent quality. Most of the effluent quality parameters are less than 10 milligram per liter. Okay, so this is the final things. I would like to consolidate everything in a single slide. So based on our objective, we can select any scheme. So if it is recycle or reuse wastewater, SPR and the MBR is the better alternative. We can produce all effluent quality within 10. The post-treatment is required while feeding water to ultrafiltration or the membrane separation modules. Okay, so if you want to reduce the operational cost, the ideal choice is the SPR. One is the operation cost is less. USP is another thing. So if we consider the space requirement, the SPR, MBR, MBBR, the space, okay, then consider the capital reduction in cost. If you want to minimize the total capital cost, then USP, ASP, and the MBBR. So based on your objective, we can select any treatment as per the energy cost, recurring, non-recurring, as well as the effluent quality. So to this, I would like to wind up. So if you have any specific doubt,
Thank you, Dr. Amal Raj, sir.
okay, welcome, sir.
Nice presentation. Probably equally, we had a feel like we all. You took us through an industrial visit. Such a, it was a very good presentation, and there are a few questions now asked from attendees side, and I think you have answered for those questions during the last few slides. But still, what I'll do is, and I mean, the attendee who has asked the question, I'll put him, he'll talk to you right now. Mr. Kottapalli Gurulingam, I'll allow you to talk now. You can ask your question to the presenter right now. Dr.
Okay,
Amal.
sir.
Yeah. You can talk now, sir. Mr. Kottapalli Gurulingam.
Okay.
I think, probably, he has got the answer over it. Then Mr. Simanchal, I'll unmute you now. You can ask your question to the presenter right now.
Hello.
Yeah, Mr. Kottapalli Gurulingam, can you ask your question, sir?
Yeah, I'm Gurulingam. Thank you for your excellent session. An insight has been given for treatment of water, wastewater. My question is, see, I have many questions, but one of them is, in the process of treating the water, there may be possibility of emerging some hazardous gases. So how to handle these hazardous
Sir, your voice is not audible. A little bit louder.
Yeah, Mr. Gurulingam, can you make yourself a little bit louder, please?
Yeah.
Still,
See,
I think,
in the process of wastewater treatment, there is a possibility of hazardous gases emerging out. So how to deal with these, how to venting them out, these hazardous gases?
Yes, okay. During anaerobic treatment, hydrogen sulfide and methane will be liberated. While constructing plant, we have to provide methane gas sensor as well as hydrogen gas sensor within the treatment plant site itself. By this way, we can sense it. Accordingly, we have to take suitable precaution. Safety measures should be provided while commissioning any sewage treatment plant, because the water producing gases like hydrogen sulfide and the toxic gases like methane will be liberated if the anaerobic system is not properly handled. Okay, so we should be very careful while operating anaerobic systems. Aerobic system doesn't matter because the carbon dioxide is evolved. When hydrogen sulfide will be liberated if anaerobic condition is established while treating the wastewater. But we should be very careful about the methane gas. Normally, it will be collected there. If any leakage is there, then the fire hazard will occur. Methane gas is a very explosive gas. It's a toxic gas. So we should be very careful. So methane gas sensors should be provided with alarm facility. So by this way, we can provide suitable safety measure within the plant itself. That is the routine way of doing while constructing the plant.
So the post-treatment,
Yes.
what would be the quality of water with respect to its pH value and hardness when compared to a potable water system?
Okay, while using for a gardening purpose, the C-pH standards are very stringent. C-pH 2012 standards are very stringent. The turbidity level should be less than 1. Okay, then phosphorus content should be less than. I think the very stringent standard, the fecal coliform level should be nil. Okay, the for irrigation standard itself. Okay, so we have to eliminate all pathogens from water and keep fecal coliform less, nil only. So by selecting disinfection is mandatory. Before that, if you want to reuse water for industrial application, RO is mandatory, reverse osmosis plant. Otherwise, for gardening purpose, we can consider ultrafiltration module, because the power required to operate ultrafiltration module is lesser than RO module. RO module normally pressure is 30 to 40 bar of pressure is required. But doesn't matter. Here we can consider ultrafiltration module suitable for disinfection. Post disinfection, it can be UV, ozone, or chlorine. So chlorine, if chlorine reacts with organic compound to form a carcinogenic substance, instead of chlorine, we are suggesting UV or ozone. That is ideal choice to reuse for flushing purpose. For industrial purpose, RO is mandatory.
Yeah, for industry, normally this softening plants, the hardness is taken care. Whether in the reverse osmosis process, the hardness can be controlled.
So it's a little louder.
Hardness of the
Hardness
water.
doesn't matter. Hardness is normally the hardness of water is in line with drinking water. And mostly we are permitting 200 is the desirable hardness for as per IS 10,500 2012. Suppose the hardness is doesn't matter. It more fine. Antiscalant should be added while installing the RO module. So while RO system is there, we should consider the hardness causing substances. So antiscalant should be added while feeding the water. through RO module. So if you want to reuse water for industrial purpose, at Chennai city we have 45 ml day TTRO plant, tertiary treated reverse osmosis system, because that is under operation. Every day we are producing 45 ml day of water for industrial purpose. Those industries are located near Oragadam, far away from Chennai city. Okay, that is our application. Because in addition to that, we can use for gardening as well as flushing purpose. So the calcium issue can be addressed by selecting antiscalants. That is inbuilt in our pre-treatment. So our well feeding water to RO model, we should consider the calcium, because the chemical fouling should be avoided. Yes, that's a very good question.
Because in a metallurgical plant for this furnace water cooling system,
Yes,
the water, the returned water is being treated and again reused. So
yes.
there, this hardness is playing a vital role in forming scales in the tubes.
Yes.
So in this RO process, can it be controlled? This is my question.
RO means, for example, if you consider groundwater as the treatment, there's permissive up to 200 doesn't matter. Suppose the hardness level above 200, the scale formation is the obvious thing. But that can be controlled by softening phenomena. Instead of RO, we can consider softener. So sodium-based exchange resin type of softener, or acidic-based exchange resin as a softener to remove hardness for drinking purpose. For sewage, there's a used water, the same, the quality is more or less similar to the drinking water. So mostly hardness of hardness is there. Hardness in sewage water is in accordance with the drinking water, because the drinking water is emerged as a wastewater with some contaminant, but the hardness will not be added during the water usage. But sometimes while using groundwater, in addition to municipal water or the metro water, we are using our own groundwater. That's why the hardness level may be slightly high.
Okay. So
Thank
my
you.
last question is about the cost effectiveness of these plants.
Sir, it's a little bit louder. Sir, your voice is not so audible.
See, my last question is about the cost effectiveness of these plants when compared to desalination plants. Can it be comparable with the desalination plants?
Yes. So desalination plant is instead of antiscalant, we can provide softener prior to RO system. The desalination plant that uses seawater only, there no need to provide antiscalant. The hardness is within the limit one, but some hardness is there. There we can provide antiscalant.
Okay.
The most of desalination plant pre-treatment to remove the suspended matter, then the disinfection facility. Then pre-treatment can be done by disc filtration followed by ultrafiltration module. Okay, then the antiscalant and pH correction. That's all. Okay, in order to avoid antiscalant should be added.
Thank you,
sir.
Mr. Gurulingam. Thank you, sir. Yeah, there is another question from Mr. Santosh. I'll unmute you, sir. You can also interact with the presenter. The question goes like this: will the treated water going out of ASP be measured for BOD? If so, what should be the limit? That's his first question. One more question from him. Okay, right. You can answer his question, sir.
Okay, definitely BOD is there. During tertiary treatment, the BOD is completely eliminated. For example, the secondary treated wastewater, the BOD content is less than 10 milligram per liter in SBR, MBBR, and the MBR. But in conventional ASP is less than 30 milligram per liter. By providing rapid sun filter and the activated carbon filter in a sequential manner, we can keep BOD level less than 10. Then another way, then the chlorine oxidation is required. Once the chlorine oxidation is over, we can completely eliminate the BOD. Then it can be further be treated membrane separation phenomena or biological nutrient system to address the residual BOD present in secondary treated wastewater. Some BOD is still there. It should be taken care by filtration and the chemical oxidation or biological, sophisticated biological system before feeding RO, because the RO membrane should be protected. Otherwise, fouling will occur, biofouling will occur. And there's a chemical scale formation is also there. That should be addressed by providing antiscalant. But here, so biofouling should be controlled. That's why the BOD should be eliminated. BOD is still there in treated effluent.
Hope Mr. Santosh got the answer.
Lesser level, only 10 milligram for some advanced treatment, but 20 to 30 milligram less than 20 for some other treatment lesser. BOD is there, but 300 to 200, 20 or 300 to 10, that's all.
Yeah, there is another question from Mr. Kapil. Kapil, you can ask your question to the presenter. some delay.
Hello, sir.
Yeah, please, Mr. Kapil Dev. Yeah, sir. It's taking some time, probably. So I think some network issue from their side. Probably it's taking some time. Any more?
Hello, sir. Yes.
Yes, sir.
What's
No. I think yeah. So
Hello.
Yeah, please.
Sir, this is Santosh Kumar.
Yes.
So my
Yes,
query
that-
for the presenter is to, sir, have you come across any plants which are working by the integration of IoT platform to the sewage treatment?
At present, we are not focusing in IoT, but in future it
Okay.
may be possible.
Okay.
Okay, I think it's a very good idea to integrate these two. Okay, it's a very good idea. But we are using the PLC and other controller to operate the system. It's not connected to IoT.
Okay.
Okay.
Thank you.
Yeah, since we are running short of time, we can wrap up the session. Once again, I thank Dr. Amal Raj for his excellent presentation on the overview of water treatment. Then he took us through various sewage treatment process, like aerobic process, anaerobic process. Then again, he took us through effluent treatment process like MBBR, SBR, MBR, ASB, and he gave an excellent comparison among all these processes and which is best. Based on our objective, we can select all those processes. And we hope from the organizing team, we hope this session was very useful for all the attendees and particularly even for the organizer. Once again, we thank Dr. Amal Raj sir for spending his valuable time and coming forward to give this presentation.